A couple things. I will post a quiz today that's endocrine and muscle, right? Because you have a test a week from today, so I'll give you some time to work on that. It's open book and all that kind of stuff. There will also be a worksheet posted that goes with muscle. I don't know if we'll have time to do it in class, so I'll post it for you. It's really all the complicated stuff that we're doing today. It kind of simplifies it so that you have a fill in the blank for all of the most complex stuff. Good? Okay. Are there any of you guys who didn't go into sarcomere structure in anatomy? Anybody? Okay, everybody did. That's great. So I'm going to assume that you know the basics of sarcomere structure. Of course, you also did it in lab a couple weeks ago. So we're going to start with the simple part of sarcomere structure, and we're going to go up from there, right? So you guys should remember, I hope, the ends of a sarcomere are called what? Z-line, Z-disc, Z-band, Z-something, right? The protein filaments that attach to the Z-lines are called what? Actin. And the ones that don't attach to the Z-lines and float in the middle are called what? Myosin. By the way, myosin doesn't actually float. When I took physiology, we're like, I don't know, it's just staying there. I don't know how, it's fine, it's magic. The answer is never magic, FYI. Okay? The answer is we haven't found it yet. But there's actually a stabilizing protein called the M line. It doesn't matter physiologically, but it does make the story make more sense, right? So the M line stabilizes it. Other things that hopefully you've learned before are that the myosin has projections sticking off, and the projections are called myosin heads, right? And the myosin heads, we will see, are going to be important today. The myosin heads are very, very attractive to a binding site on actin. Unfortunately, people are not very consistent about what they call these two things. So I'm going to be consistent. I'm going to call the things sticking off of myosin heads. Okay? And I'm going to call the things that it binds to on actin active sites. Some textbooks call them myosin binding sites, which is a little confusing because you're naming one protein with something. It's like, this is the zygomatic process of the temporal bone, the temporal process of zygomatic bone, and everybody wants to flip you off. Okay. You're like, I don't know how you did that. That was rude. So here is what I am going to do. Today, they're going to be myosin heads and actin active sites. The metaphor that I use for them quite often is hands that grab handles. Okay? With me? So, we will see that when the myosin head is bound to an actin active site, it's called a cross bridge. Okay? And we will go through cross bridge formation and cross bridge cycling in detail today. Okay, so let's talk about stuff that you have done in anatomy and how it's related to this. And this is the way I told you I was going to start this lecture, which is you learned in anatomy the basics of sarcomere anatomy and the basics of sarcomere contraction, I think. I think that what you learned is that the reason you have to learn the anatomy all the way down to the submicroscopic level is that this is the basis of contraction, right? Because when the myosin heads reach up and grab the actin active sites and pull, what happens to the distance between the sarcomeres? Shortens, exactly. And if you shorten enough sarcomeres, you can shorten a whole myofibril. Shorten enough myofibrils, you can shorten a whole muscle fiber. Yes? Shorten enough muscle fibers, you can shorten a whole fascicle. Shorten enough fascicles, you can move a bone. Yeah? Okay, so now let's look at the banding pattern. And of course, you guys remember that skeletal muscle is striated. Now, the structure of the sarcomere has to do with the striations, right? So wherever you have myosin, because it's thick and dense, it is going to be darker, and you call that the what? A band, yeah? Wherever you have myosin but no actin overlap, it's going to be dark but not as dark, and you call that the A zone. And then wherever you don't have myosin, you're going to be called the I band, right? Okay, so the question I asked you on the video was, which of these is going to change length upon contraction? A band, I band, H zone. I band, yes? And H zone, yes. Okay, so I always tell this story because I don't, maybe it was like this with you too, because She took animal physiology for her first degree. I took animal physiology. When I was taking physiology, I'm way older than her. When I was taking physiology, we actually did animal experiments in lab. And I realized that that's ethically a little iffy. However, I will tell you that, that I will never forget what I learned when I anesthetized a frog, took out his gastrocnemius, stretched it across a microscope slide, and then stimulated it with an electrode. Because what I saw under the microscope slide is that before it was stimulated, it was striped. After it was stimulated, it was almost completely dark. And why is that? Because everything pulls into the A band, right? And then I would unstimulate it, striped, stimulate it almost completely dark, right? And I literally never forgot that. And it's not like you guys are not going to forget whatever the hell you did on iWorks. Let's be honest, okay? You're like, I don't even know what that was. I don't even know what happened. Okay. So I'm not saying everyone should do that. However, I do think it's a really important thing to understand. So you can see the changing of the banding patterns that occurs with contracted and relaxed muscle. Okay. So now what we are going to do is we're going to add detail to this, right? Because there's more to it than what you learned in anatomy. And then after we add detail to this, we're going to go back and hook it up to the nervous system and figure out how you get all this started. Good? Okay, so basically what we are going to start talking about is we're going to focus on a single cross bridge and look at what happens at a single cross bridge. You guys with me? A single cross bridge. And what happens at a single cross bridge is called cross bridge cycling. So orient yourself to make sure that you can figure out what we're looking at here. So what we are seeing here is these little black dots right there are the actin-active sites, yes? And this is the myosin head, right? Okay. When an actin-active site is bound to a myosin head, it's called a cross bridge. And what we are going to see is that cross bridges cycle. So here's what was kind of implied. We implied if you reach up, hands reach up and grab the handles and pull, you shorten the thing. But that's not what actually happens. If you look at what happens at a single cross bridge, you have to ratchet it. You have to go bind, release, grab the next one. So on each side, you're going, and you're ratcheting the whole damn thing in, right? Because remember that although we represent this as two-dimensional, it's not two-dimensional. The sarcomere is actually deep. Yes? Okay. So that means that even if I let go of a single cross bridge, is the thing going to completely be released? No. Because I've got multiple holding on at the same time with depth and with length, right? Okay. So I can ratchet the damn thing in. So basically imagine that, do you guys remember the monkey bars when you were little? Yeah. Only I want you to imagine a big, beefy-ass kid that is so big, he sits in the middle, and he pulls the monkey bars toward him. Right?
That's scary.
Yeah. All right? Both sides. Right? And so he's pulling it into him. He is the A-band, right? And that's his nickname, A-band. Okay, you with me? All right. So now I want to teach you cross-bridge cycling. So when we do cross-bridge cycling, where are we? Okay, I'm going to teach it to you 2 times. The first time I'm going to go through and just show you how the cross-bridge moves. The second time I'm going to put in the energy so that show you how the energy is required for cross-bridge cycling. Okay, and you guys are going to have to clap with me. Ready? Okay. All right. Everybody hates it when I do this, but guess what? They also wrote it on my evaluations that I wish you would make me wiggle more, even though you all give me dirty looks when I make you wiggle. You do remember things. Okay? All right. So here's how cross-bridge cycling is going to go. Ready? Okay. This is cross-bridge formation. Just reach up. Hands reach up and grab the handles. Okay? This is called the power stroke. Okay? And now I'm stuck right here. And then I'm going to have to release the cross-bridge and reposition. And then I have to do the whole thing over again. Cross-bridge formation, power stroke. Release, reposition. Formation, power stroke. Release, reposition. You guys are fantastic. Okay, so do you understand what's happening at a single cross-bridge, right? Okay, so why is it called cycling? Because you really don't do any appreciable amount of moving with moving at once, but it's always going to release, reposition, grab the next one. Release, reposition, grab the next one. Does that make sense? Okay. Now, Where would you think that ATP, which is the source for cross-bridge cycling, would be used in this whole process?
You think that with the power
So I would think power stroke. That's intuitive. But if I give you a different metaphor for it, you'll understand why it's not the power stroke. This thing works like a catapult. So now I'm going to ask you again, where do you put the energy in for a catapult? Yeah, to position the catapult, right? Right? You put it in the high energy position. That's how you get it to work, right? And then once you've got the catapult in the high energy position, all you have to do is release the pin, and then the catapult goes, okay? So now I'm going to go through once and add in the energy, right? Where the energy is used, and then you guys are going to flap for me again. Okay, so now I want to show you that when I start at position 1, which is the binding of actin to myosin, I already have used ATP there. Used ATP is ADP plus an inorganic phosphate. Yeah? That means that the ATP has already been hydrolyzed. Big burst of energy has been released, right? So it's going to be started at position 1 here, okay? So position 1, I'm going to bind. That's free. I don't have to pay for that at all. Actin and myosin love to bind to one another. Okay? Then how do I allow for the power stroke? Pull the pin. The pin is inorganic phosphate. As soon as the phosphate falls off, yes, I get my catapult release. Power stroke. Yes? Okay. Now I'm stuck here unless the ADP falls off, right? And makes room for ATP to bind. So 2 things have to happen. 3 is ADP falls off. 4 is binding of ATP. The binding of ATP is what allows you to release actin from myosin. You break the cross bridge. Yes? But I'm still stuck here. How am I going to re-energize my catapult? Hydrolyze ATP back to the original position. Yes? Hydrolyze ATP. All right, one more flappy. Flappy, flappy. Okay, ready? All right, now we're going to add in the energy. Okay, so cross-bridge formation. Yes? Does that cost us anything?
No.
No. I'm stuck here unless what occurs? Phosphate falls off. Phosphate falls off, and that allows for what to occur? Power stroke. Okay, I'm stuck here unless 2 things occur. Give them to me in order. ADP falls off. I'm still stuck here. Yes? ATP has to bind. What does ATP binding do? Release of the cross bridge, break the cross bridge. I'm stuck here unless I do what? Hydrolyze ATP back to the original position. Yes? Does that make sense? Okay. Yeah, hydrolyze ATP. When you hydrolyze ATP, you break it down to ADP and phosphate. Does that make sense? Okay. That's the hydrolysis reaction we talked about at the beginning of the semester. Add water, break it down to ATP and phosphate. Everybody good? Okay. So that is cross-bridge cycling. Now, I want you to extrapolate to how freaking energetically expensive muscle is. Because this is one cross-bridge, right, that is going to go multiple times. And every single cycle requires 1 ATP. But now please remember that every sarcomere has multiple cross-bridges. And every sarcomere is also not just 2-dimensional, but 3-dimensional. Yes? And now there's a whole bunch of sarcomeres in a myofibril, and a whole bunch of myofibrils in a muscle cell, and thousands to 10,000 muscle cells in a single muscle. And so skeletal muscle is incredibly energetically expensive. And it's really more energetically expensive than you think, because we generally think our skeletal muscles are only using energy when we're wiggling. But I want to tell you that skeletal muscle has a baseline level of contraction called tone, right? So it really never goes down to low energy consumption, which is why if you have a boatload of skeletal muscle, what can you tell me about your metabolism compared to somebody the same height, same size, who doesn't have a boatload of skeletal muscle? Incredibly energetically expensive, which is probably why Sasha can eat a lot more than I can. Right? Can you eat a lot?
I can't eat a lot.
Yeah, she's a power lifter. So lots of skeletal muscle makes your body really, really energetically expensive. Does that make sense? So you can have a male and a female, for instance, who are about the same body size, but typically speaking, because of testosterone, males are going to have more muscle mass than females, and that's why my damn husband can eat so much more than I can, and it's not fair, and I would like to speak to the management because he doesn't even like food half as much as I do. But he's got a whole bunch of skeletal muscle. He's also 6'6", so that helps too.
Is he a picky eater?
What?
Is he a picky eater?
No. He just thinks food is an inconvenience, that like it has to be done. It takes so much time. And I'm like, I have literally been thinking about the next meal in the middle of this meal. Right? And how when we go on vacation, he's like, I really want to go see this. I'm like, I have to eat this? I eat this? I eat this too. I mean, all of it. And he's like, do you do? I was like, that's why I travel. I have to eat things. Okay. So you guys all right with cross-bridge cycling? Okay, so that's cross-bridge cycling. Let's show you, I'll wait for that animation for just a second. So the notes are the length of the sarcomere shortens, but the lengths of the filaments, the protein filaments, don't change. Hence it is called the sliding filament theory of muscle contraction. Yes? And we thought this was happening before we actually had the capacity to stain the proteins 2 different colors and actually confirm it. So we looked at this pattern happening under the microscope, and we're like, hey, I think that what's happening is they're sliding past one another. And we're like, that sounds like a good theory. And we called it the sliding filament theory of muscle contraction. And then we got the ability to stain myosin one color and actin another with fluorescent staining proteins, and then you can actually confirm that that's what's happening. Does that make sense? So sliding filament theory of muscle contraction. So let me just show you briefly to confirm what you guys told me. When you have sarcomere shortening, you can see that the A band doesn't actually change, the H zone and the I band will change. Ready? Wiggle, 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 wiggle. That's the sound effect. When it's maximally contracted, the H zone can completely disappear. Good? And then it's going to relax back to its original position. By the way, for those of you who care, not that it's particularly important, but they did find another little protein right here called titin. This protein called titin is attached to the edge of the myosin, and it's like a spring. So if you completely let it go, it'll like spring back into position. Yeah.
Is that essentially what it's for then? Like help
Yeah.
spring back?
Mm-hmm. Yeah, but we didn't know about that one when I learned this either. Like these two are new. So probably within the last 15 or 20 years, those have been discovered. Okay, so now I want to talk about how you hook this up to the nervous system, okay? Because here's the thing about skeletal muscle. Skeletal muscle really doesn't do anything unless you directly tell it to do something. So skeletal muscle actually needs to be stimulated directly by the nervous system. And specifically, we know enough anatomy to know that it has to be stimulated by the somatic motor division of the peripheral nervous system, right? Okay. And generally speaking, skeletal muscle contraction is usually voluntary, not exclusively, but usually voluntary. So the somatic motor division is often called the voluntary division. But we know, of course, skeletal muscles doesn't always behave voluntarily, right? Otherwise, you would forget to breathe. Right? Because have you ever been so busy that you know you would forget to breathe if it was the extra thing on your plate? You're like, I literally would have died yesterday if I had to remember to breathe. Okay, so what we will see is, just going back to a little anatomy for you, is that, for instance, the skeletal muscle, if I were trying to contract my biceps brachii, the motor neuron cell body is probably in the cervical region of my spinal cord. Right, in the gray matter, the cervical region of my spinal cord. What I actually send out through, ultimately, the brachial nerve, right, is going to be just the axon, right? Action potential down the axon will cause neurotransmitter release, right? The neurotransmitter release is going to be how we get here. I just need to put in the in-between portions between the neurotransmitter release and how you actually cause sarcomere contraction. Is that okay? Okay. All right, so putting the A together with the P. So now what I'm going to tell you is the important part is the site of neurotransmitter release and communication between a neuron and a skeletal muscle fiber is called a neuromuscular junction. It's just a type of synapse, right? So you know about synapses from anatomy. We talked about them briefly here. This site is going to be important because it is going to be the site at which you stimulate the skeletal muscle fiber with the neuron. Now, just to reinforce, the thing that is important for you guys to understand is that skeletal muscle doesn't do a damn thing unless you tell it to. And you have to be very specific. You have to tell every single skeletal muscle fiber to contract with the nervous system or else it won't. So its default is to do nothing. So for instance, if I were stimulating Stasha and you guys were cardiac muscle, I would only have to tell her and she would tell all of you guys to contract, right? Because cardiac muscle has what? Gap junctions. Skeletal muscle doesn't have them. So if I was stimulating you and this one got broken, she would do nothing, right? The metaphor I always use is like my teenagers, right? So teenagers don't do a damn thing unless you give them specific instructions about what to do, right? So if, for instance, I ask my son, he's a delightful human, he is also an adolescent. If I ask him to take out the garbage, but I do not ask him to replace the liner in the can, what is the probability that the liner in the can will get replaced? Like 0, right? Because their objective is to do as little adult things as possible so they can go back to doing tween things, right? Okay? So, skeletal muscle, teenagers, right? You have to tell every damn one of them, every damn time, every damn instruction, right? And then only sometimes will they actually do it. Okay, so you have to tell them, and I love them. And I was the same way. It sounds like I'm being superior. I was the worst teenager. I owe so many apologies, just retroactively. I'm like, oh, maybe I just won't ever talk to any of those people again. That'll be better. Okay, that's healthy, right?
That's where you moved, right?
Right, that's why I moved, right? I get to be myself, and people are not like, oh, you're the one who didn't like tie your shoes for 4 years. I'm like, no, that wasn't me. Okay. Oh, God, yeah. I was the most bookish, weird child, right? Yeah, and I like to argue with my teachers a lot, too. So that was fun. You can't? They were wrong. But actually, in retrospect, they weren't all wrong. Okay, so, all right, so skeletal muscle fiber must have a direct neural stimulus to contract. So now let's start with how you get that going. Okay, so we have this portion called membrane excitation. So here is the deal. We have to start with the nervous system before we get the membrane on the skeletal muscle fiber activated. You with me? Okay, so you have to start with the nervous system portion. So let's go right here. So this one talks about it in a whole bunch of detail. Let's simplify it a little bit with this one. Okay, this picture is really busy. It's also awesome. So later on, when you are studying, look at figure 12.8. But let's just concentrate on a part of it at a time. The first thing that happens is, of course, you get an action. This is my cell body. This is my axon. This is my axon, telodendria, and axon terminals, yeah? So I get an action potential that is generated on the somatic motor neuron that causes an action potential, and then it actually causes release of the neurotransmitters from the synaptic terminals. Now, luckily, it's always the same neurotransmitter. It's always acetylcholine, always. We have never found an exception to this, not only in humans, but in all mammals, all vertebrates, which means this is probably really, really old, right? Evolutionarily, this is probably really old. So what happens is the action potential is actually going to cause neurotransmitter release. And it's going to go into the synaptic cleft, right, which is that space between the neuron and whatever it's communicating with. It's going to diffuse across the synaptic cleft, and it's going to bind to acetylcholine receptors on this super curved portion of the sarcolemma. Remind me why it's so curved at that particular place. Yeah, more surface area, more receptors, faster response, right? Right, right. So more surface area gives you more responsiveness because you've got more room for receptors. What is this region called? That's called the motor end plate. Okay, keep that in mind because we're going to play with that a little bit today. So that's called the motor end plate. So here's something else that you need to know. These acetylcholine receptors are also ligand-gated sodium channels. So if you bind to and open them, what is sodium going to do? Rush in, blank polarizing the sarcolemma, depolarizing the sarcolemma, and if you hit threshold, then you will get an action potential. Yeah, same story we've heard before, only we heard it in neurons before, right? So we can get an action potential. So you guys with me so far? So if you hit threshold, you will get an action potential. Threshold is slightly different in skeletal muscle than it is in neurons, but it's still the same direction. Yeah, it's the same direction. It's depolarizing. Okay, but the action potential is also anatomically different in muscle simply because it doesn't have to stay on the surface. The reason it doesn't have to stay on the surface is that there are tunnels of plasma membrane, of sarcolemma, that go deep into the cell. So it goes along the surface, both directions, because the dominoes are set up so that they'll go both directions. But then it also goes down deep into the tunnels, right? And those tunnels are called T-tubules or transverse tubules, right? And remember that they looked like this, right? You're right. And remember that they also run right next to the lateral sacs of the sarcoplasmic reticulum, right? All right, so we will get the action potential to run not just along the surface, but also down the T-tubule. And then what's going to happen is this is going to eventually get us to where we need to go because, go ahead.
Sorry,
Just
the stats, when I was listening to your pre-lecture and
Okay.
used the diagram, I'm not understanding exactly what the lateral sacs are. I see them right there. So where those holes are, where is the actual sac?
This part right here of the SR is the lateral sac. Well, it's the lateral portion of the SR, the sides of it, that has a little storage place for calcium. That's why they call them the lateral sac. So this part right here is the lateral sac. I'll show it to you down here too.
Openings where you can see the yellow part.
No, this is the lateral sac
Right.
of the SR.
After purple.
Yeah, and it wraps all the way around the myofibril.
That's the sac.
Yeah, well, not this portion. This portion would be just your regular SR that probably would be doing lipid synthesis.
Right.
This portion right here, right next to the T-tubule, is storing calcium.
The green part, correct?
No, the green part is the T-tubule.
Purple next to the green.
Purple next to the green.
Okay.
Purple next to the green is the lateral sacs of the SR. Go ahead.
Reticulum means network.
Yeah, it does.
So when the sacs are the ones that are sandwiching the green parts, and then the
This is the reticular part.
Yeah,
This is the netty
network.
part.
Yeah, that's how the calcium is traveling through to get to the sarcolemma.
Yeah. Okay, so now I want to talk about how this will allow for calcium release and what calcium release has to do with this story. Okay, so what's going to happen is if I have an action potential, meaning I depolarize to threshold, meaning that, of course, the nervous system just stimulated me, or I would sit there, I would be sitting there doing nothing, like a teenager, right? Just sit there doing nothing. So now what's going to happen is the action potential is going to go down the T-tubule, and it will cause the release of calcium from the SR. Now, I don't hold you responsible for the mechanism that it causes calcium release, because when I learned it, it was another thing that was magic. They're like, oh, and it just like gets out of there. I don't know. It's fine. But I will tell you that now we know. What happens is the T-tubule actually has two linked proteins, one that's from the T-tubule and the other that's to the SR. And by the way, do you remember the SR is actually just a folded plasma membrane, right? So they're two separate plasma membranes. Somebody had to figure out how the hell they activate one another. What happens is you get an action potential to go down here, change the shape of this protein so that it actually knocks into that protein and opens the protein right there. It's called a ryanodine receptor. And a DHP receptor. I will never ask you about that. But the two are linked, and basically an action potential causes this to be released from the SR. Okay, it opens up a calcium channel in the SR. Now, calcium is stored in the SR, yes? So as soon as I open up a calcium channel, calcium is going to move out of the SR by what transport mechanism? Yeah, just diffusion, right? High concentration, low concentration. And it's going to move into what fluid? Extracellular fluid or intracellular fluid? Intracellular fluid, not extracellular fluid, because remember we are still inside the cell, yeah? Right? And it's going to dump a whole bunch of calcium right near where it needs to be, because these guys were wrapped around the myofibrils, right? Is it all going to be directed to each sarcomere? No. You're just like dumping it, right? But you're dumping way more than you need, yes? So you dump way more calcium than you need, and then what will happen is calcium will find a place that has a calcium binding site. And the calcium binding site is on one of these little proteins associated with actin. So let's show this, and then we'll show what the impact. Okay, this is before you tell the cell to do anything, right? This is after the action potential and the calcium release, yeah? Calcium will bind to troponin. Troponin is the switch that operates the barricade protein tropomyosin. When calcium binds to troponin, tropomyosin will move out of the way, and then I can see these gorgeous, attractive actin active sites, which myosin loves. You've just been keeping us apart, stupid troponin, right? And now troponin got out of the way, and our love story can become requited. So calcium is going to bind, yes? And then now the actin active sites are exposed, right? And then now we start flip-flopping, right? Actin active sites are exposed. For cross-bridge formation, tell me what happens next. No. Phosphate falls off. Power stroke. Now what? ADP falls off. ATP binds, release. Hydrolyze ATP, do the same thing over and over again. Yes? Does that make sense? And that will go on for as long as you have ATP and calcium present. Yes? Good? Okay, so everybody all right with that? All right, so let's look at an animation, and then we'll talk about how the hell you get this thing to relax. Okay, so wait, let me make sure I covered everything in the notes. So we got the membrane excitation, the nervous system portion, the muscular system portion, the contraction portion. Okay, so now we're ready for... All right, so now we are going to look at these two. So, okay, so these two are kind of linked to one another. So what we are going to have is the neuromuscular junction. I'm going to get an action potential that goes down the axon of the somatic motor neuron, just using all the terminology for you. You guys don't know about that channel yet. I'll teach it to you in a little bit. Okay. I am going to have exocytosis of the neurotransmitter, which is what? Acetylcholine, always acetylcholine. Acetylcholine will diffuse across the synaptic cleft and bind to an acetylcholine receptor that is also a what channel? Ligand-gated sodium channel. Yes? And then sodium will rush in, blank polarizing the cell, depolarizing the cell. If you hit threshold, which is represented here by a flash, because they didn't want to draw in all the voltage-gated ion channels, but there's a whole bunch of them, right? So it's represented by a flash, right? Then what's going to happen is you are going to get your action potential that moves down T-tubules, causes calcium release from where? Sarcoplasmic reticulum. The colors aren't the same because it's from a different textbook, but it wasn't that color anyway. So, so now I'm going to get calcium release from the SR, right? And watch calcium go find its binding site, right? Nope. Okay, so calcium's going to bind to what protein? Troponin. And move what protein? Tropomyosin, yes. And then as soon as tropomyosin is out of the way, your love story can be requited. Troponin and actin and myosin will find one another, right? So I am going to do, come on, get up there. Cross-bridge formation, phosphate falls off, power stroke, right? And they only show you one. Is that good? Okay. So this is putting the whole story together. What did you say, Charis?
Well, right before which, where did the phosphate fall?
They don't have it in this figure.
Okay.
This is just simplified, so they didn't put in the energy required. We know it falls off before you actually do the power stroke, right? None of these animations has everything in them, just none of them. Go ahead.
Right before the power stroke or whatever, so we were talking about earlier how, like, you know, they during the power stroke, they don't all do it at the same time. So, like, if one lets go, it's not going to go rebound.
Right, because there's hundreds of them by length and also hundreds of them by width, right? So are they all going to be in exactly the same timing? No, because it's just random how calcium finds, right, the troponin and moves it out of the way, and that's when you actually form the cross bridge. So in all likelihood, all of those random events are not going to be perfectly timed, right? That makes sense? They're just not going to be. So it's you are not going to let them all go at one time until you force relaxation. And we're about to talk to you about how relaxation occurs. Okay? Go ahead, Charis.
Does that have to do with that varying times where they're all contracting? Does it have to do with, I don't know how to word this, when you're lifting something and you have to sustain that and hold that for a while, if they were all going up once, it just doesn't.
That's a different thing. This is in one cell, alternating cross bridges.
Okay.
You're talking about in a whole muscle alternating motor units, and we're going to get to that
That's what
next
I'm
time.
thinking, motor units.
Yeah, that's like alternating motor units firing so that you can have sustained muscle contraction. Yeah, that's a gross function, and this is a microscopic function.
Got you.
Okay, all right. So now I want to talk about relaxation. Now you need to understand how relaxation occurs so that you can understand a couple of different disorders. Okay, so the deal with relaxation is actually an active process. It actually requires ATP. So let's talk about how the big picture of relaxation is going to have to work. So in order for relaxation to occur, guys, does it make sense to you that you better stop stimulating the neuron with the somatic motor neuron or the axon? So as long as you continue to stimulate it, right, and release acetylcholine, you're going to continue to depolarize it and potentially get action potentials and contraction, right? So the first thing that has to happen if you ever want a muscle to relax is, well, stop contracting it, right? Either voluntarily or involuntarily. Just stop sending it from the nervous system. So the action potential on the axon has to end, yes? But then after the action potential on the axon has to end, you have to deal with the acetylcholine that's there from the last time you released it, yes? Okay, so the acetylcholine is there, and as long as the acetylcholine is there, it's going to continue to find a receptor, right? And continue to depolarize, right? So you have to get rid of the acetylcholine. The way that you get rid of the acetylcholine is that on the postsynaptic cell, on the motor end plate, in addition to having your voltage-gated sodium channels, you've also got an enzyme present, and that enzyme present is called acetylcholine esterase, which sounds really long, but it just is the word acetylcholine, the word erase with st in the middle, right? Acetylcholine esterase. Okay, so let's look. So we saw that in this figure. Well, we didn't actually see it because I didn't play it for you yet. So now I caused my action potential, and now I'm done, right? Now what I have to do is I have to use this enzyme called acetylcholine esterase. What it does is it breaks acetylcholine up to acetic acid and choline, and it reuptakes the valuable one, which is choline, which is a B vitamin derivative, so that it can use it again to make more acetylcholine, right? But if you break it in half, is it going to activate the receptor anymore? No, because it's shape specific, right? So it just pops it apart, right? And then reuptakes the part of it. Go ahead.
So it has to diffuse twice because it has to go through the cleft and then into the vesicle, right?
Into the vesicle?
Yeah, like so.
It doesn't actually diffuse. By the way, the reuptake mechanism that they're not showing is it's a pump.
Oh, okay. I was like,
There's a pump there. Yeah, they just don't show it out there. What?
it's reabsorbed into the neuron?
Yeah, it's reabsorbed into the presynaptic cell.
So choline
Yeah.
is reabsorbed into the cleft.
Choline is
And then
reabsorbed.
Acetic acid is then bound from there and then they go to the vesicle.
No, acetic acid just diffuses away, and you just make some more acetic acid.
You make it with the choline.
Yeah, you reabsorb the choline. Choline is a B vitamin derivative, so it's like the limiting factor here. They don't reabsorb the acetic acid.
Become acetylcholine, it goes back into the vesicle.
It goes back in, and you actually remake it.
In the cleft?
No, they remake it inside the presynaptic cell.
Sorry, the knob, I mean.
Yeah, inside the presynaptic cell in the synaptic knob, yes.
Okay.
Yes. Uh-huh. Okay, we haven't talked about neurotransmitters in detail yet, so this is a little unfamiliar, but we will talk about all of these things in more detail in the next set of notes. I had to do muscle first because try to make it align with your lab, even though I think it makes more sense to do neurotransmitters first, and I think they should have rearranged the lab, but they won't listen to me. Okay, so the enzyme that you need is acetylcholine esterase, right? And it's present on the postsynaptic cell, right? On the sarcolemma on the motor end plate of the postsynaptic cell. Okay, so now here's what we just accomplished. We just stopped stimulating it, and then we just took care of the acetylcholine that was there from the last time, right? And so without an action potential, or without a depolarizing graded potential, I shouldn't get an action potential, right? But then, of course, we have to take care of the calcium that was released from the last time. Because as long as calcium is around, guys, what's going to happen is these guys will continue to find their actin active sites, right? So I have to get rid of the calcium. But I'll go ahead and tell you that boatloads of calcium is stored in the SR and you didn't release all of it. So how do you get it to go back where it came from? It diffused out, but it ain't going to diffuse in. You're going to have to pump it. Yeah. So this is a pump that is on an organelle, which is probably kind of unusual. But it is. It's an SR pump. It pumps it from the sarcoplasm, the intracellular fluid, back into the SR fluid. Yeah? Okay. And it's active. So if you don't have ATP, I want you to think about what that means for the muscle, right? Not only does it limit contraction, but it also limits relaxation. Yes? Okay. So you have to pump calcium back in. And as you pump calcium back in, the tropomyosin slides back into position at certain little places, right? After you pump all of it back in, then it all slides back into position. And after it all slides back into position, your tighten will allow you to relax again. Yeah? Will pull the sarcomere back to its original position. It will relax back to its original position. Does that make sense? So after tropomyosin is blocking the actin active sites again, you will relax back to the original position. Is that okay? Okay, so what we will see here, so relaxation. You got to get rid of the action potential, then you got to get rid of the acetylcholine, yeah? And then you got to get rid of the calcium, and that's done by pumping. ATP is required from there. After you get rid of the calcium, the tropomyosin comes back into position, and you force the actin and the myosin to let go of one another, right? And it's really the tropomyosin falling back into position that like it breaks the cross bridges. Actually, it's the ATP that breaks the cross bridges, and then the tropomyosin prevents the next one. And then the sarcomere will stay relaxed until another action potential arrives. Good? Okay, so go ahead. Till the neurotransmitter does it, all starts again. All right, so let's reinforce this by talking about when it goes wrong, okay, to make sure that you actually understand it. So you guys know what rigor mortis is? Okay, so let's talk about what rigor mortis is and why it's occurring. What is the problem with being dead?
No energy.
No energy. And why is there no energy? No oxygen. It's primarily oxygen is the issue. So no oxygen, right? No oxygen after death because you're not breathing, which will make you run out of ATP really quickly, which will end up doing something that's kind of counterintuitive. It's counterintuitive that your muscles actually stay contracted, and they actually build contractions after you die. They don't do this, right? But what they do is, in whatever position you died in, two things are going to happen. Remember how you release your cross bridges? You have to have ATP to release your cross bridges. Eventually, you are going to run out of it, and you won't be able to release your cross bridges. Simultaneously, you are going to be leaking calcium down its electrochemical gradient from the extracellular fluid into the intracellular fluid, and from the SR into the intracellular fluid. And that means that more tropomyosin is going to be moving out of the way, and I'm going to be forming more cross bridges. So it's not as if I stay paralyzed in whatever position, right, or whatever level of muscle contraction. I actually get more contracted after I die because of the calcium leakage, right? Calcium leakage in combination with no ATP means I'm going to form more cross bridges. Now, it's not enough to wiggle me. I mean, there are a couple situations in which you can do that. That's not this. But it is enough to allow me to be in sustained muscle contraction in whatever position I died in. Yeah? If you leave me there for a few hours, I will build up rigor in that particular position. So if you look at this guy, you can tell that rigor has set in, because if you were a living person, how long could you hold that position? That is so hard to hold, right? You can hold it for, 30 seconds, a minute, 2 minutes, and then you would be screaming, right? You would be yelling at people, right? Because it's all abs, back, and thighs, and glutes, holding on to that. Okay, so this is rigor, and rigor is primarily due to the combination of calcium leakage and the lack of ATP, right? And so when will rigor leave the body? When your proteins start to degrade. Because your protein degradation will occur, right? And protein, as soon as your actin and your myosin start to fall apart, they can't hold on to the cross bridges anymore. And when does that occur? Well, that's totally temperature dependent, right? You guys know that you'll go through all of the stages after death really quickly at higher temperatures and really slowly at lower temperatures, just like every metabolic reaction, right? So if you die in a trailer in the summer in Texas, which was literally the reason I got degrees, so that would not happen to me. That's like the whole reason I went to college, right? Because it's like, that was the default in my family, right? In the summer in a trailer in Texas. what is going to happen is that you are going to go through all the stages super duper quickly, right? It's going to be really, really hot. If you die in the summer in the trailer in Texas on the toilet, that was that 3rd degree. You want to make sure. What happens is you die and you stay in whatever position you died in, and that means it's going to be really hard to get you out of it because your muscles are not going to let go. So in a trailer bathroom, they got to get you out. And if you're also fat, I need another degree apparently to make sure that doesn't happen. If that occurs, your muscles are not going to let go. They're probably going to have to break your bones to get you out. This is important for people who deal with people immediately post-mortem, EMTs, paramedics, all of those kinds of people, coroners, because you can't just leave them there, right? You can't say, I'm really sorry that your family member died, I'll be back in 15 hours. So you sometimes have to break bones to get them out or cut the frame of whatever you're trying to get them out of. This ends up being culturally relevant. In some cultures, people are required to be buried within 24 hours, which means that you are going to have to deal with people in rigor. Does that make sense? Now, obviously, I'm a cultural idiot because I'm from Texas, but I do know that that is, in Orthodox Judaism, they have to be buried within 24 hours. Anybody else know any other cultures that require that? They have to be buried within 24 hours and no embalming. What?
In Islam.
In Islam, 24 hours?
That's what, I know from previous people, but I'm not 100%.
I mean, it's different. I mean, depending on which sect you are, or whatever religion or faith you have, it may be a thing or it may not. But in Orthodox Judaism, you have to be buried within 24 hours and you can't be embalmed. So you have to deal with that kind of stuff with rigor. Okay, so that's rigor. Let's talk about a couple more. I want to give you guys a think, pair, share question. And then we're going to go through all of the other disorders. And I want you to really think about how this disorder would impact your muscle. And then we'll do it again. Okay? You ready? I got to get into your class first, though. 4 or 5. What?
Yeah, it's the same. That's what I thought. I just wanted to double check.
It's temperature dependent. Oh, 24 hours and it's some.
That's what I thought, but I just each side is different. So
Yeah. Okay, try this one. See if you can figure out the impact of this disorder. I'll tell you the 2 important parts are autoimmune and motor end plate. There are a lot more people here than there were in the first class. First class only had like 20 people. Nine more people. 8, 7. One more person. There we go. Okay, so let us go through the other disorders, and then I want you guys to think about myasthenia gravis a little bit more. Okay, so let's talk about a couple of other disorders that have to do with skeletal muscle. Okay, polio. Let's talk about polio, also called poliomyelitis. So you haven't experienced polio because there has been a polio vaccine for decades that was so incredibly effective that it almost completely eradicated polio from the planet. It still exists in a few little pockets and pops up every once in a while. I would be willing to bet that within the next 10 or 15 years, we will have to be vaccinated for it again because of the tragic privilege of the Western world choosing not to vaccinate against things. So what polio does is, polio is a virus that destroys somatic motor neurons. The virus infects and completely destroys the somatic motor neurons. Somatic motor neurons are amitotic. So is this temporary change or permanent change? It's permanent. And of course, the skeletal muscle will not contract unless it's stimulated by a somatic motor neuron. So it causes skeletal muscle paralysis. And then, of course, muscle wasting. As you don't use them, the muscles actually diminish in size. And then, of course, you start to lose bone mass. Yes? Because if you don't use your bones, then they will also diminish in size. So this is permanent paralysis of this little boy because of polio or poliomyelitis. I think this is a fantastic picture. This dude is an activist who had polio. This is polio, vaccinate. And this makes me think that we would like each other. 0.0. That's some funny crap right there. You like him, don't you? I'm like, could you be my grandpa? Because I totally like you. So polio causes permanent paralysis. People ended up on iron lungs because of polio. We had one of our presidents had polio. Do you guys remember who? FDR. FDR had partial paralysis because of polio. He was always photographed standing up, though, because height, people equate it with power, and whenever he was meeting with other heads of state, he would always be photographed standing up. But he had partial paralysis because of polio. So that's polio. Cramps. What is a cramp physiologically? It's something specific. Cramps are action potentials on motor neurons that fire at abnormally high rates, usually because of ion imbalances, usually because of the ones that you would predict, either sodium, potassium, or calcium, or a combination of them. So if you are having abnormal amounts in or abnormal amounts out, right? Diarrhea, diuretics, all of those kinds of things, throwing up, you can have cramps associated with it. Hormone imbalances can also cause cramps, right? And the one that will get you is that stress hormone, aldosterone. So why are cramps sometimes associated with stressful times? Because of aldosterone release. Remember that aldosterone gets released whenever corticosterone gets released? And so it can cause pH imbalances and sodium and potassium imbalances. So if you have cramps, you probably won't know why, but eat yourself a banana, have a glass of milk, and take a damn nap, and they'll probably diminish. Or maybe not. I don't know. Okay. Hypocalcemic tetany, tetanic contractions because of hypocalcemia. Remember we talked about hypocalcemia and how calcium was the latch on your sodium channels? Yeah? And when the latch is gone, cells will depolarize when you didn't mean for them to and get inappropriate action potentials. And you can get into tetanic contractions. This is also that hypocalcemia, what leads to those eye twitches that make you crazy and nobody else can see them, right? Usually that's a calcium deficiency. Don't know always that it is, but drink a little milk, right? Whatever you do, and it'll usually go away. Okay, muscular dystrophy. Let's talk about this one, and then we'll do the others, and then you guys can do myasthenia gravis. Muscular dystrophy is a genetic disease. It is, most forms of muscular dystrophy are X-linked recessive, which means, based on your biology, that you know which sex is more likely to manifest. Yeah. Males, yes. Okay? Now, what?
Girls can still get it.
They can still get it, but they would have to have a double dose since it's a recessive, right? So it's really rare, unless, of course, you had inbreeding or something, right? Yeah, mom and dad would both have to have it. It's just, it does occur.
At that point, it's 25%, right?
It's 25% probability with a female. So males are usually the ones who get this disease. Males only have one X chromosome and they show everything that's on it, right? Everything that's on it is there. So this is a progressive degeneration of skeletal and cardiac muscle fibers. Here's what happens. You have a weak cytoskeletal protein because of a genetic mutation. And so as you continually pull on skeletal and cardiac muscle fibers, you break them down. It leads to a progressive deterioration of the muscle. And there are, of course, a couple of muscles that you can't not use, yes? Your diaphragm and your heart, you can't stop using them. So a lot of times these little boys will die because of asphyxiation or cardiac arrest as that happens. Does exercise help this situation? No, exercise is going to worsen the situation. And there are some treatments for it. There aren't any cures yet. There are some genetic testing. You can figure out whether you actually have it or carry it. A lot of times what happens is they're diagnosed relatively young. They don't usually pass it down simply because their life expectancy is not such that they're usually going to be reproducing. So little kids, when they start to crawl and walk, they have these kind of patterns for pulling up that you guys have probably seen, even if you didn't notice it. So they start to crawl, right? But then when they get to the walking stage, when they start to pull up, they really try to like use their legs, right, to like help them, because your legs are usually really strong, right? And so they're pushing themselves up and crawling up. So if your legs are really weak, what happens is you tend to use a little bit more leverage with inanimate objects, and you pull yourself up that way. And that's one of the first triggers that would tell a physician that's paying attention that we might need to test for muscular dystrophy. Does that make sense to you guys? It's a different gait pattern and a different walking pattern. They also have some weakness in their legs that makes them walk different when they start walking. Okay, we'll come back to myasthenia gravis. Let's talk about a couple others that are related to muscle. Tetanus. How'd your mom tell you can get tetanus?
Getting stuck with a rusty nail.
Stuck with a rusty nail. As usual, she is at least partially right. Okay? It doesn't have to be a nail. It doesn't have to be rusty. There is a bacterium called Clostridium tetani. It's a pretty ubiquitous bacteria. It exists all over the damn place. And if you get it into anaerobic conditions, it produces a neurotoxin. And what the neurotoxin does is causes problems with acetylcholine being removed from the neuromuscular junction. And then you go into tetanic muscle contractions. Does that make sense to you? You'll go into what we call spastic paralysis. Flaccid paralysis is I'm relaxed and I can't contract. Spastic paralysis is I'm contracted and I can't relax, right? Either way, you can get into these situations. Now, this can be and is deadly. Doesn't kill everyone. But if your diaphragm paralyzes, your history. And here's the important thing for you guys to understand about it. It's the neurotoxin that kills you. It's not the bacteria directly. So even if, for instance, I had the tetanus neurotoxin released, antibiotics isn't going to do me any good at that point. Yeah, because it's the toxin that's already into your bloodstream, right? Luckily, there's a vaccine for this, okay? And the vaccine for this keeps you from getting the disorder. And you have to redo this vaccine. This one only lasts for a little while, every 10 years. And since you guys are going into healthcare, you're going to have a tetanus vaccine. And you're going to have it regularly. You're going to have to stay up on your tetanus vaccine. But tetanus is really only a problem with a certain type of wound. Really, it doesn't have to be a nail and it doesn't have to be rusty. What it really needs to be is a wound that closes on itself and creates anaerobic conditions, like a puncture wound, right? And it can be a splinter. I know of somebody who died because of a splinter in their knee when they were gardening. That's how they died of tetanus. Yes. And she was 45 and got tetanus.
Was it in Texas?
Yes. Huh? Oh, I didn't hear. You got it everywhere. You guys have it.
I'm just saying.
What?
How quickly do you have to be treated?
You will die within 72 hours.
So
Generally
did
speaking.
she just wait and not go?
She went, but the toxin was already in her bloodstream. But how quickly would you go for a splinter in your knee? Right? Right? When she started having symptoms that couldn't be explained by the splinter in her knee, she went. She died. She actually lived for a little while because they kept her on a ventilator. But her respiration was shut down within 72 hours. Oh, yeah.
Like just from being cut.
Yeah. So this is tetanus. So really the deal is that a closed wound, if you have a deep wound that closes on itself, then you've got the bacteria in an anaerobic condition. That's the kicker, right? That's why puncture wounds are a big deal, right? A scrape is less likely to cause tetanus than a puncture wound that closes on itself, right? Yeah? Okay, so that's tetanus. And then botulism. What'd your mom tell you about how you get botulism? Canned food. She was partially right. So Clostridium botulinum. Go ahead, Piper.
I have had botulism. So I had it 3 years ago from beets. So be careful with like putting it in soil. And my friend put it on a barbecue, and it was very hard to diagnose.
Oh my goodness! I'm so happy that you're here. So Clostridium botulinum. Here's the deal. This bacteria, same genus, different species, so they have a decent amount in common. This one will also produce a neurotoxin when it's in anaerobic conditions, right? And so what happens is, let's say you're canning food at home. So were they home canned?
No, she put them in tinfoil, make them anaerobic on low barbecue heat, and then put them on the counter.
Oh my God, that's some crap luck, Piper. The hell? Wow. Okay, so if you're canning food, this is how most people get this. You're supposed to boil the hell out of the jar and boil the hell out of the lid. Yes? Okay. And then you're supposed to put whatever food you're going to put in there, heat the hell out of it to kill off all the bacteria. And then you're supposed to create a vacuum, right? But the vacuum is an anaerobic condition. If you didn't do steps 1, 2, or 3 properly, then you may have bacteria in there, right? So why the can bulging? Because bacteria will produce carbon dioxide as a byproduct of their metabolism, which can tell you there's some critter living in there, right? So if you open something and it hisses at you for like half an hour, throw it down the damn sink and don't even feed it to your dog, okay? Right? I don't take home canned food unless I was involved in the canning process, right? I ain't taking it. I will say thank you. I'll write you a thank you note, and then I will pour it down the sink and think, oh, that looks so good.
That's why you're supposed to, like, when you use the canned pasta sauces, you're supposed to use all of it. In one go. Like,
Interesting. I don't do that.
Storing it is not a good idea.
Interesting.
Don't even open it.
No, don't even open it. Oh yeah, spores. So what botulism does is causes a problem with preventing the release of acetylcholine from the neuromuscular junction. So this causes flaccid paralysis, not spastic paralysis, but flaccid, right? And so you're relaxed and you can't contract. It is also, though, the basis of Botox cosmetic, and also the basis of the clinical uses of Botox. But let's talk about the cosmetics uses of Botox. So I definitely don't want botulinum toxin in my bloodstream because I'd be dead, right? Or at least in a really, really hard way. How long did it take to go to that?
To what?
To get over it?
About a month and a half after I had antitoxin. My eyes were just, I couldn't even open them and I had double vision.
Oh my goodness. Wow.
I broke and made it, couldn't even talk.
You're the only person I've ever met who has been a Botox or a botulinum toxin survivor. Okay, so you don't want to inject it into your bloodstream, and I would bet that she doesn't get it injected into her face. Probably not, but I do. So if I don't want these, all I have to do is paralyze these muscles. But I want those, I have to paralyze those muscles, right? Okay, I haven't done it in a while. So what you can do is you can go to a physician who's been to medical school. Do not go to the woman who's doing it at a wine party and is also drinking the wine, who's a nurse, right? I want somebody who understands skeletal muscle anatomy, right? I also want somebody who understands the physiology. So what they will do is they will inject it locally, and what it will do is when you inject it locally, it will actually destroy the somatic motor neuron's ability to release acetylcholine at that location. As a matter of fact, it actually eventually the axon terminals just degenerate, right? And so those will stay gone for about 4 months, and then you will regenerate the axon terminals, and then the muscle contraction will come back, right? So if you can't contract the muscles, you don't get the wrinkles, right? Also, if you like paralyze them too much, then you don't get any facial expressions. You're like, oh, that was fantastic. This is amazing. I love that. All right? So you have to be careful. So they can also use this for actual clinical uses and not just cosmetic uses. Migraines, yeah. It works well for some people who have migraines because the muscle contraction in the neck actually is the precursor to the migraine, and if you can paralyze some of the muscles in your neck, you can reduce the likelihood of migraines. They also use it for hyperhidrosis when people are sweating entirely too much, like sweating so much that it's really like impacting your quality of life. They try to use it for the hands, but there's so much innervation here that it's really difficult and requires lots and lots of injections. And then it only lasts 4 months, and you got to do the whole damn thing again, right? So lots of sensation, lots of injections. And then this is the cool one. The guy who does my Botox is actually a reconstructive surgeon. And you can use Botox if you had injured your face and you had asymmetry. If, for instance, this side is drooping, I can paralyze this muscle so they're at the same level. So if you had a person with a really, really, really good knowledge of anatomy and facial anatomy, then they can actually like allow for more symmetry in your face by doing unilateral Botox injections, which is really cool. And he showed me a bunch of his pictures with like facial reconstruction. Like a lot of how your face appears is due to your muscular contraction. So that's Botox. Last two, and then we'll do the think, pair, share. And that is poisons. Okay, super nerd kid, read a whole bunch of murder mysteries as a kid. One of them we learned about curare. I learned about curare from Agatha Christie. Oh my gosh, you're such a nerd, I love you, you're my favorite. Okay, so curare is a poison that's derived from a South American plant, historically used on blow darts and arrows and whatever the hell, pointy things, so that you could eat a critter without having to chase the critter. You want to eat the monkey, you don't want to crawl up and get the monkey, you boom, and the monkey falls out of the tree. What it does is it blocks the receptors on the sarcolemma, right? And so the muscle completely relaxes and it'll kill you because of the diaphragm being paralyzed, right? You can still eat the critter because it's got a really, really short half-life. Yeah. So they would take critters that they didn't want to chase and they would just dunk them and then they just wait for it to fall down and then go get it and eat it later. We also use it to kill each other because humans don't like other humans that much. So that's curare. The other one, some nerve gases and pesticides, and I think this one's Zyklon B, although I can't quite remember. I should have written it down there. I knew I'd forget. Pesticides aren't meant to do this to humans, but nerve gases we designed specifically to do this to humans. Most nerve gases that we are still using or have been used in war situations were created by Americans. Yeah, we made most of them, and a lot of other countries actually got them. We even sold them to other countries, historically speaking. So what does nerve gas do? It actually inhibits acetylcholine esterase. So what would happen? You can't relax. So you breathe in, and you're stuck. And that's the last time you breathe. Does that make sense? So that's how some nerve gases work. They don't all work the same way, but that's how some of them work, and we have used them.
So
okay.
Consider a suffocation then? Is that
Yeah, you will suffocate. Mm-hmm. You suffocate with curare too, but you suffocate with curare because your diaphragm can't contract, and with nerve gases because your diaphragm can't relax. Yeah? Okay, so now you guys, I hope you have all the tools to talk this through with your buddy. You ready to do it again? Okay. Again, the important parts are autoimmune and motor end plates. Talk amongst yourselves. Make sure that you're talking to someone.
What the hell?
8 people left. Seven people. One person. 5, 4. Get your answer in. 3, 2. There we go. Okay, guys. What do we think the answer is?
B.
The answer is B. How'd you guys do? 62%. Okay. So now I want somebody who chose B to talk me through why that one. Somebody talk me through it. Who? Jane. Okay. Let's do
So
it.
the motor end plate is the part that's attached to the muscle and it's made of a cell membrane, right?
It is made of cell
So
membrane.
it can be repaired.
Good. It
So,
can be repaired.
If you manage the symptoms with medication, you can put yourself into a type of remission where you can live.
Or if your immune system
manageable-
just stops being a jerk.
Yeah, that
Yeah.
too. But
Mhm.
sometimes
So why do you know that it leads to partial or complete paralysis instead of sustained muscle contraction or anything? Who wants to do that part? Chairs, go
Would
ahead.
it have to do with muscle wasting if the transmitter is not getting through to activate the muscle?
Uh-huh.
Then you'll have some muscle wasting and.
Well, wasting is later. Initial stages are just either spastic paralysis or flaccid paralysis. Which kind is this one?
Isn't it because, so it can't start the action?
Exactly. Can't start the action
There's
potential.
no- Yeah, it's losing
Right.
activity.
Does that make sense?
So it'd be weak, it wouldn't be.
Be
So
weak,
would that be
right?
flaccid?
Yes, that's flaccid paralysis. I didn't use that term, but just so you know, there's 2 different kinds of paralysis. Neither one of them get a damn thing done. One of them's floppy and the other one's spazzy, right? Flaccid paralysis, right? Or spastic paralysis, right? Either one. So this one would be flaccid paralysis, in which you wouldn't be able to contract partial or complete. And apparently, would you tell that your experience with the patient that you're taking care of? She's taking care of a patient right now with myasthenia gravis.
So when she first came in with it, she was so weak. I mean, she's still, because she's in remission right now, but she's still so weak that sometimes we have to hold her head up and things like that for her, because before, like, she couldn't close her eyes, she couldn't hold her head up, she couldn't move her arms. I mean, even, like, because she had to be ventilated, so she couldn't talk. And so she couldn't even write anything out because she was so weak, she couldn't even hold a pencil at first.
So what do you give her?
They did plasmapheresis.
Yeah, she asked me why that worked, and I have no idea, so I wrote it down, and I'll see if I can figure it out. Or maybe they just do it, and it works, and you're like, I don't know. Whatever. I mean, there's an awful lot of that in medicine. You have no idea how much. Like the whole lithium to treat...
Somebody replace your plasma?
yeah, but I have no actual knowledge about how that works, but I will look it up.
Certain cancers too.
I know, I don't know what the hell it is. Go ahead.
I've had it.
You've had...
All my antibodies out, my injection.
Uh-huh.
So they took my spleen out, and then they did plasmapheresis so that my antibody levels would go back up, so it could have.
Ooh, I wonder if, maybe it was an antibody treatment to take down her immune
That's what I
system.
was thinking. I was like, it's basically rebooting.
That makes sense. Look at how smart we are collectively. Oh my God, we're so brilliant.
All pieces together.
I know. Okay. All right, we're going to switch gears. This part's not hard. So I just want to talk about skeletal muscle metabolism and how skeletal muscle gets all of this ATP that it needs for both action potentials, right? And cross-bridge cycling, and then also relaxation. So it's incredibly energetically expensive. We know that. And the source that it uses is ATP. And so skeletal muscle needs a boatload of it. And, of course, your skeletal muscle has to be really immediately responsive, right? No lag time because you'll get hit by a bus. So the deal is that skeletal muscle has quite a few reserves to get at access to ATP. So this is mostly the story that you've learned before, but this is just showing it to you in skeletal muscle. We know that this is aerobic cellular respiration, right? Okay, plus a couple of extra things that other cells can't do, like creatine phosphate. I'll teach you guys about that in just a second. And lactic acid, which can allow you to sort of redo glycolysis when you run out of oxygen. Okay, so, but this is basically skeletal muscle. So I want to talk about how skeletal muscle gets immediate access to ATP, the next thing that it gets access to, and the next, right? So there's a whole bunch of reserves. So first thing is, don't you get your first power stroke for free because you already paid for it, right? So as soon as you've had an action potential, that first power stroke was paid for by the last round of ATP, right? Right? You already put the catapult into high energy position. So you form this, the phosphate falls off, and you get that for free. But of course, that's not even seconds of activity. You know, that's whatever seconds, milliseconds, I don't know, I just made that up. Less than a second. Okay, what next? What do you use for your next quick access to ATP? A conversion mechanism that is a way faster access to ATP than all of these biochemical pathway reactions. So the deal is, inside skeletal muscle fibers, you have some creatine phosphate stored. And what creatine phosphate is doing is holding on to a phosphate that it can very, very quickly give to ADP to make it back into ATP, which is faster than doing all these damn reactions, right? Okay. So this is like a second, right? You don't have to do very much. All you need is creatine phosphate, which skeletal muscles store, and an enzyme called creatine phosphokinase or creatine kinase, which skeletal muscle also has. You ready? Okay, so what's going to happen is right after I did this, the very first access to ATP, if I'm starting from 0 ATP, would be this, right? I'd take the used ATP from the last time I did something, right? And I would rephosphorylate it, and it would give me ATP enough for about 1 minute at a brisk walk or about six seconds at a sprint, and then I would be out of it, right? That make sense? Okay, so the enzyme needed is CK or CPK. We'll talk about how it can be used clinically next time. Okay, so now I'm like 6.5 seconds in, right? Or maybe a minute, right? Maybe a minute maximum into muscle contraction. Now, I want to start using the oxygen that was bound to myoglobin, simply because my cardiovascular and respiratory systems may not have caught up yet. Because you start wiggling before you tell your cardiovascular and respiratory systems, right? You just start doing it, right? So how can I make sure that I have enough oxygen? Well, I stored a little bit of it, so I can make sure that I've got enough oxygen for aerobic cellular respiration. I didn't need it for this, right? Now I'm going to need it for aerobic cellular respiration, and it was on the myoglobin, okay? And I am going to be able to use that stored oxygen for a little while. A little while is dependent on you and how much you had stored. But now I can also, that myoglobin, I also stored glycogen, right? So I had oxygen stored on myoglobin. I had glycogen storing glucose, right? And of course, I got mitochondria. So even before my cardiovascular and respiratory systems catch up, I can do a little bit of aerobic cellular respiration. How much is a little bit? Well, quite a lot, depending on how well you fed yourself recently. That breakdown of glycogen and the stored oxygen, and also the oxygen that you bring in. Your breakdown of glycogen can last 5 to 10 minutes. Just the glycogen that was in your skeletal muscle. It depends on how hard you're working. But it can last 5 to 10 minutes. Now, by that time, of course, your cardiovascular and respiratory system would have caught up, right? Okay, which is good because you're going to run out of glycogen in a second, right? So I'm not saying that the oxygen lasts 5 to 10 minutes, but the glycogen quite often does, okay? So now, you are going to be using this for the first 5 to 10 minutes. So now we're like 11 minutes in, okay? And then hopefully my cardiovascular and respiratory system have caught up to bring me enough glucose, right? And then you'll start burning blood glucose, yeah? And also using oxygen that you brought in from your respiratory system. Everybody good with that? And then that will last most of us, and I'm almost done, most of us up to 30 minutes. So now I'm 41 minutes in. Yes? 41 minutes in on average. And then, and only then, have I run out of enough resources that I will start to burn fatty acids. Does that mean you have to work out for 41 minutes before you start burning fat? No, it means that your skeletal muscle has 41 minutes of stored nutrients that it can use. But do all of your tissues? No. All you have to do to burn fat on any given day is wiggle more than you chomp. That's literally all you have to do. But your skeletal muscles are not going to switch over to fatty acids for a while because they have made sure that they've got all of these resources stored. Does that make sense? Okay, so we will pick that up next time and we'll talk about lactic acid and muscle exhaustion next time. Okay, I'm going to post the endocrine and muscle quiz for you. Review quiz, right? I'll post it today. We won't be finished with all the muscle stuff. And I'm going to post a worksheet for you. And the worksheet will just be sort of a simple way to do a fill-in-the-blank review of all the muscle contraction and relaxation stuff. Okay?